Us again. Like Colette said, my name is Marija Kvas and I'm Senior Application Specialist here at Separations. And today we will be talking about RNA applications. For those of you that joined us last time, you will know that we were talking about microbes, how to identify, characterize, and monitor them, either if they are infectious diseases or the good guys. But today we will look into different RNA applications and how we can use those to improve the human health. Why is NGS a solution? When we spoke last week, we talked about whole genome sequencing, whole exome sequencing, many different applications. But because we have such a sequencing, we can sequence so much deeper. We can generate vast amounts of different data. We are able to uncover rare and complex variants. And at the same time, we can look at many different samples. So not only do we have a lot of data available to us, we can do it across multiple different samples. And that ideally allows us to have that qualitative information where we not only look at changes in DNA, like we look at SNPs or insertions, deletions, duplications, inversions, but we can also look into transcriptomics and how does that RNA change, either looking into gene expression or gene fusion, alternative splicing, but we are allowed to look into that pairwise differences. And it allows us to actually do, like I mentioned, gene expression and to look not only what is happening in the host, but also to look into host and pathogen interaction, and also to see maybe how is, for example, drug response, how is a certain treatment being affecting the host. Before we dive into different applications, let's go a little bit back. Let's go into back to basics, onto the central dogma. All of you know that all of our cells have DNA, and that DNA is transcribed into mRNA, which is then translated into different proteins, which we need for building blocks of the cell or for the different processes in the cell itself. And while DNA is constant, and all our cells have the same DNA, the amount of RNA in our cells is always different depending what are the cell's needs. Some genes are on and off, and mRNA might actually not be present for that specific gene. And also depending are those genes up or down regulated, we will have different amounts of RNA in the specific sample. So if we look into DNA, like I mentioned, it tells us about identity. It's always, we have always same amount in the cell. It's always telling us the same thing, unless we have cancer, but it's always constant, right? That DNA is always stable. But RNA is indicative of activity, right? That is constantly changing. It's constantly responding to environment, to stimulant. So it's different at all the times. It's very dynamic, right? And obviously varies from one cell to another. So when we're talking about RNA, obviously there's different classes out there. We briefly mentioned about protein synthesis, right? Where we have, you are familiar with messenger RNA, ribosomal RNA, transfer RNA. We need all these three in order to create proteins. But we can also talk about co-transcriptional modifications and post-transcriptional modifications. And in that case, we're talking about small nuclear or nucleoral RNA. And then we can also talk about regulatory, where we have microRNA, small interfering RNAs, and so on, right? When we are working with RNA, we need to be mindful of a couple of RNA features. We need to be mindful of what is their size, right? Different RNA types have different size, like our microRNAs are very small, about 20 base pairs, where our messenger RNA, mRNA, is about 100 to actually 1,000 base pairs long. Obviously, then we need to understand the shape. How is it folding? How is that RNA folding over? Is it forming secondary structures? We need to understand the stability of that molecule and how long is it present in the specific cell. Also, when we're talking about RNA extractions, and also we need to know how much is present in the cell. What is the copy number of that of the specific RNA. So when we talk about NGS, obviously, like I mentioned before, the advantages to NGS when it comes to RNA-seq, obviously we have enormous discovery power because we don't have to have the prior knowledge of what we are sequencing. It's hypothesis free, right? We can discover novel transcripts, for example. We can look into different variants, and we do this with a high specificity and accuracy, right? And these applications have a huge impact on oncology, on genetic research, on infectious diseases, like, microbiology. So even though today I will focus mostly on human RNA and applications around human as a host, obviously, all living organisms will have, if they have DNA, they will have RNA, obviously. So any of these applications can be applied to many different organisms out there. So when we're talking about different applications, I mentioned gene expression profiling, where we're quantifying the coding transcriptome to understand the biological state. We can talk about transcriptome discovery, where we are looking into novel features, like novel transcripts, where we're looking into biomarkers. And we can also look into transcript detection, right, where we are detecting RNA isoforms, SNVs, genes fusion to expand understanding of disease risk and status. And because obviously we're doing this with NGS, we have that qualitative and quantitative data. We have a novel insight for any actual species. We don't have to have any prior knowledge, and we have a high resolution and sensitivity. Again, why would we do genome expression? We need to understand which genes are active and how active, and actually when they are active, depending on maybe different treatment, different drug that we are inducing. So we need to understand what is physically active, what is on, what is off, right? But we also want to not only understand coding regions, sometimes we are interested also in the non-coding transcripts, and we are always interested in novel transcripts, novel gene fusions, alternative splicing, right? And this allows us biomarker discovery, validation of variants in tumor normal genome, and so on. Okay, so when we talk about RNA workflow, all of it, I would say it's same. Obviously, we will start with RNA extraction. We will do RNA QC, which is really important. And then depending on the library prep, actually, what is our application, we will go into a library prep. We will quantify that library. Depending on the amount of data that we need, we will choose a specific sequencer that we need, and obviously we would analyze the data. So when we talk about RNA extraction, remember I said we need to understand the stability of RNA, because RNA is easily degradable. So when we work with RNA, we need to be extra careful. We need to store it at -80. When we're talking about RNA QC, obviously, we are not just interested in concentration of RNA. Yes, it's really important to us, but more important is the purity of that RNA, so that we don't have anything that will inhibit our reaction downstream. But also we want to know the integrity of RNA, right? How degraded it is. And when we're talking about RNA experiments, we're talking about what we call a RIN value, RNA integrity number, and there we want it to be anything about 8. I'm talking here about obviously mammalian RNA. It might differ for different species, but like I mentioned, we're going to focus on human today, mammalian hosts, right? So that integrity of RNA is really important. And we also wanted to have it DNA treated, right, so that it doesn't interfere going forward into the library prep. And like you noticed, depending on what is our biological question, right, if we are interested in just coding or both coding and non-coding transcripts, we will choose a library prep, and we will go into a little bit more depth there. Obviously, once we have the library, we want to make sure that it's a correct size, that it looks good before we go into sequencing and data analysis. So like I mentioned, regardless of what library prep you choose, first of all, we need to purify that RNA, right? Majority of RNA in our sample will be ribosomal RNA, so about 80–90% of our RNA is ribosomal RNA, which doesn't really tell us much, right? We are interested in coding regions in mRNA, right? So we have two options to remove that rRNA. We can either deplete it, right? We physically pull it out, or we can use poly-A selection, which will capture mRNA, right? And then from there on, we will go into obviously cDNA synthesis. So we will do a reverse transcription where we would create double-stranded cDNA synthesis. And then, like any other library prep, we want to ligate the adapters onto our cDNA in order to make it sequencing ready. This is a really simplified workflow, but in a nutshell, that is the principle behind the library prep. So when we talk about capturing mRNA, we're talking about pulling it from poly-A tail. You will know that eukaryotic mammalian mRNAs out there are actually processed, and that the mature mRNA will have 5' methylation cap, and at the 3' end will have what we call a poly-A tail. And if we leverage on this poly-A tail, and we have oligo DT beads, so poly-T sequence that is bound to the magnetic bead, this poly-T will actually bind to that poly-A tail, and we can pull that mRNA with a magnet, right? But if we are working with FFPE samples, which are usually degraded, right? If we employ this kind of methodology, we will create 3' bias towards that poly-A, right? Because our DNA will be fragmented, and we will only be pulling actually the poly-A tail. So it's not ideal when it comes to FFPE samples. In that case, we will employ what we call a Ribo-Zero, which is ribosomal RNA depletion method, right? And historically, Illumina had probes that would bind, magnetic probes that would bind to our RNA, which we will pull it out, right? But recently they came out with a simplified approach, more target-specific approach, where we have DNA probes which will bind to the specific, they will hybridize to the specific rRNA targets. And then we will have the enzyme that will recognize this DNA RNA complexes, and which will actually cleave it and digest it, right? So we have enzymatic depletion of rRNA, right? And that allows us not to have any accidental carryover of rRNA into a library prep, right? So also what is very nice about this new Ribo-Zero Plus is that in one tube, we are targeting multiple rRNA species, not just cytoplasmic and mitochondrial rRNA and globin transcripts, but we're also looking into bacterial rRNA for gram-positive and gram-negative bacteria. So we are actually depleting all of these rRNAs. And because we are pulling out the rRNA, what we are left with is processed and unprocessed mRNA. So that's why we are looking into coding and non-coding regions. And like I mentioned just now, after having that RNA purification, where we remove ribosomal RNA, we focus on mRNA, we will reverse transcribe that into cDNA, and then we will continue with the library prep. So just a brief summary of library preps, or actually applications, RNA-Seq applications that we have out there. If we are working with totalRNA, then we are talking about global expression profiling, non-coding RNA discovery, alternative splicing detection, biomarker identification. If we are only looking into mRNA, which is just our coding transcripts, obviously we're looking into differential expression profiling, transcript level abundance, allele specific expression, isoform identification, and fusion discovery, right? And then I will touch briefly later on a targeted approach when we are only interested in a specific allele expression, right, into specific fusions. We are looking at a specific targeted approach. So we'll touch on that briefly. Illumina's power is in this integrated sample to answer workflow, where obviously we have different solution for our library prep. We have a different sequencing systems out there, and Illumina also have a base-based sequencing hub, which is a cloud-based analysis server that has over 90-something different applications out there, and within those is also DRAGEN RNA and DRAGEN RNA differential expression, which allows us to analyze our RNA data. So when we're talking about Illumina sequencers, this is what we have in Illumina protocol. And for those of you who joined us last week, where we talk about small genomes, microbes, your viruses and bacteria, certain small fungi, we were talking about these benchtop sequencers, right? So the sequencers that have a smaller capacity, because we are working with the small genomes, we don't actually need to produce a lot of data, therefore we can do it on the smaller benchtop sequences, right? But when we're talking about RNA, we need, when we're talking about human genome, we have over 20,000 genes, and those genes being expressed, right? Not all of them at the same time, but it's a huge amount of data that we need to generate, right? And as such, it's not cost effective to do it on the benchtop sequencers. We need to employ those production scale sequencer. And you will see that we will talk about NextSeq 550, NextSeq 2000, and the NovaSeq, because they have that much higher capacity of producing large amounts of data and being more cost effective. We will put any RNA-Seq experiment or NGS project onto these larger production scale sequences. When we talk about NGS, you usually talk about coverage, but we talk about coverage when we talk about whole genome sequencing, right? When we're talking about sequencing DNA, right? Because that DNA is constant, right? It's always the same. But RNA is dynamic, it changes all the time. So it doesn't really make sense to talk about coverage. Here we're talking about the depth, how much have we sequenced, right? So when we're talking about RNA sequencing, we are talking about number of reads that we have sequenced, right? And obviously, deeper we sequence, more confident we are at what we are calling. So if you're looking at the gray area, this is our reference genome. And if we had just one read, we wouldn't be confident in that variant, is that variant true variant, or is it maybe because of the sequencing error? But if we are sequencing deeper, if we have more reads, right, then we can with confidence say that, yes, that is a true variant, right? And because genes are present at different copy numbers, also we need to sequence deeper in order to kind of, when we sequence and count and normalize this, we need to understand what is going on and how, with what confidence we can call this, right? This graph here is just to illustrate, if we have lower amount of reads, right, we would think that they are maybe less expressed. But if we have a higher increased number of reads, then those lower expressors, actually we can realize that they are not lower expressors, right? So it's really important that, based on the application that you have, based on biological question, and also consulting with literature to understand what is the depth of sequencing, how many million reads you would need for your RNA experiment. So when we're talking about mRNA sequencing, where we're talking just about coding genes, you will notice here, like I mentioned, we will talk about NextSeq 550, NextSeq 2000, and NovaSeq. You will notice that on different types of flow cells, we can put different amounts of samples. When we talk about mRNA sequencing, we recommend 25 million reads, but like I mentioned, this is dependent on the species that you're working with. So it's really important that you consult with literature before you decide on what is the amount of data that you need. When we're talking about total RNA, because we are looking into coding and non-coding transcripts, we need almost double the amount of data, sometimes even more than that, right? But let's at least start with double amount of data, so 50 million reads that we need per sample. And that would mean that we can load less per flow cell, right? This is just, I won't go into too much details, but this is just to show you that different instruments have different capacity. You will notice that NextSeq 550, the maximum we could do is 400 million reads. NextSeq 2000 is 1.2 billion reads, and NovaSeq is 20 billion reads, right? That's why with a NovaSeq, we can multiplex up to 384 samples. So we can really have a big study when it comes to having the capacity to actually run that big study on the NovaSeq, right? To touch a little bit on RNA library prep, those of you that worked with RNA previously, you are familiar of what we call a TruSight RNA, either mRNA or totalRNA. So that's a legacy library prep. TruSight is all the kit. It's still on the market, but Illumina is always about improving and getting better kits out there to our customers. And obviously, it's not just about high performance, but also easy of use and how fast it is to prep the library. Nobody wants to spend 2 days in the lab prepping a library, right? So Illumina came with the innovative way of making a library prep easy to use, more faster, still having that high performance that TruSight library prep had, but now working with lower input samples, which is really great because for certain applications we can go as low as 1 nanogram. Previously we could do it at 100 nanograms or from 100 to 1,000 nanogram. Now we can go as low as 1 nanogram depending on our application. Maybe 10 nanograms for coding transcriptome, or even 25 nanograms. But we have much lower input with this library prep. And like I mentioned to you, we have multiple species for our RNA depletion. And what is really nice as well is that we are having ability to multiplex more samples, right? With the TruSight, we were limited to maximum 96 different samples that we could do on the flow cell. Illumina has made this library prep. They designed it in such a way that they can now multiplex. The way we add the adapters onto our cDNA has changed in order to allow us to multiplex and to have that capacity of doing 384 samples, which makes sequencing more cost effective, right? Because if we can load more samples onto one flow cell, then that becomes cheaper per sample, right? And it's all about being cost effective, right? And like I mentioned, this workflow is much quicker. We're having multiple species for our RNA depletion, and we can do much higher through output, obviously depending on the instrument that we have, right? So you heard me talking about total RNA, right? Here, like I mentioned, we're talking about coding and non-coding regions, right? Because we are depleting ribosomal RNA, we're pulling out ribosomal RNA, we are left with everything else, right? That's why it's coding and non-coding. So we're physically throwing away, well, in this instance, we are enzymatically chewing it up. We're chewing up our RNA, but everything that we are left with is coding, non-coding RNAs. And that is ideal for global expression profiling, non-coding RNA discovery and characterization, alternative splicing detection, and biomarkers identification, right? And that would be our Illumina stranded total RNA prep, right? Then we have mRNA if we are interested in looking only in coding genes. So we would employ those poly T beads to pull poly A tails, so those mature mRNAs. Everything else we throw away. So we are only left with mature mRNA, which will then be translated into a protein. That's why it's a coding transcriptome. Then we can look into differential expression. We can look into isoform identification and allele specific expression, but we can also, if we sequence deep enough, right? If we sequence really deep, we can look at the detection of lower expressed mRNA. If we are looking at the specific targeted regions, right, we are not interested in the whole transcriptome, right? Only a few specific genes that we are interested in. We want to know what is the level of that specific allele expression. Then obviously we will have a targeted approach. We can do that either by enrichment, where we would have probes that will bind to that specific specific transcriptome, and we will pull that out. So we will pull out what we are interested in. Everything else would be washed away. Or we could do it amplicon based. We would amplify the transcripts that we are interested in. Like I mentioned briefly, Illumina has, they have improved on the RNA library prep so that we have a lower input amount, right? And that the turnaround time or the hands-on time on the actual library prep is much less, right? This is just to illustrate to you that obviously depending on different instruments that we have, we can multiplex many different samples. Obviously depends on the application as well. But what I want to point your eyes towards is actual the analysis portion of it, and you will notice that when we have a non-DRAGEN analysis out there, it will take much longer to actually analyze the data, where with DRAGEN we significantly decrease the amount of time needed for the analysis. Remember, DRAGEN is that dynamic read analysis for genomics pipelines that is ultra fast way of analyzing data, and this is available on the base space. So let's look at into example of the specific workflow, right? If our study goal is to focus on coding transcripts and quantifying gene expression with multiple abundance values per gene, or to identify novel transcripts isoforms, we will obviously employ mRNA library prep, right? And we mentioned that we will look into 25 million reads, because mRNA, when we prep it, it's really short. We don't have to employ a long read length, right? Anything between 2 times 75, so per end, either 75 reads or 75 bases or 100 bases will be enough. And if we do this experiment on the NextSeq 2000, you will notice that we can multiplex up to 40 samples. And again, I just want to show you that on DRAGEN, to analyze 36 samples, it will take 30 minutes, which is really ultra fast. If our study goal is to study coding and non-coding RNA, and to analyze abundance values for every transcript, for every gene, right? And to identify novel transcript isoforms, gene fusions, and allelic specific expression, obviously in that case we need to look into total RNA, right? And remember I said, because we are looking into both coding and non-coding, we're going to need at least double of amount of data. And if we need double amount of data, then we can put half of the sample on that same flow cell, right? On that same instrument that has the same capacity, right? Same flow cell has the same capacity. Because we need more data, we can do less samples. And again, I want to show you that DRAGEN is ultra fast, and in 30 minutes, we can actually analyze 8 samples. So I will touch briefly on RNA-Seq workflow on base space. I'm by no means a bioinformatician, so I won't go into too much details, but I just want to make you aware that on base space we have DRAGEN RNA-Seq sequencing workflows out there, and obviously it will start with your FASTQ files, and we will align and quantify those reads, and then you will have a transcript level quant results and gene level quant results, right? And then we can go from those files. So from that initial analysis where we aligned our reads, we can go into differential expression, right? And from that, there is an app specifically for differential expression, which is based on DE-Seq2, and there we can look at, which genes are, what is the abundance of different transcripts, and which genes are expressed or not, right? So when we talk about differential expression, we're talking about quantitative comparisons of different genes of transcripts. So we are physically counting between 2 or more samples, or actually group of samples, right? So in that experiment, we can have different conditions, right? Either treatment, we're looking at how is a cell reacting, which genes are expressed, right? What is the abundance of those transcripts for that specific treatment, right? So we are looking to identify differentially expressed genes at the different conditions, right? And this, if we're doing this, then we can discover what is the effect of that treatment, right? We're looking into gene expression levels. We're looking into isoform abundance ratios, alternative splicing, right? So this is what differential expression allows us to do. And it's really easy to actually launch this application on base space, right? You will obviously have to predetermine what is your control, right? And then you will have to define which samples belong in which different conditions, right? Different treatments, and you will, for those of you that are using base space, you will notice that it's really user friendly. Bioinformatics friendly pipelines out there where you just have to select options and define certain for differential expression, you will have to define the comparison groups that you're working with, and you will launch that application. And like you saw, it will take 30 minutes to actually analyze 36 different samples. So very, very ultra fast. Okay. We also have availability of calling fusions, right? So we can also detect different fusions. And this is really important when it comes to cancer research, when we're looking at, not just alternative splicing, but also, gene fusions to understand which transcripts have fused together, right? And we can obviously not just detect the fusions that are known out there, but we can also look into detect novel fusions, right? When we are talking about more targeted approach, remember I mentioned we have enrichment-based approach, and here is two examples of that enrichment panels where we're talking, and this is specifically for those oncology studies that people are interested in, but we're talking about TruSight RNA pan cancer, and where we are looking at gene fusions and expression of 1,385 genes. So we are looking at about 21,000 different exonic regions, right? We are not interested in everything, but only specific gene fusions and expressions. And when we're talking about TruSight RNA fusion panel, we are talking about a subset of that of the same pan cancer, but here we are only focusing on the gene fusions, right? And here we are looking into 500 different genes, and we are looking over 7,000 different exonic regions. When it comes to targeted approach, we also have what we call RNA prep with Illumina RNA prep with enrichment. This is fairly new library prep. Illumina has borrowed on Nextera technology here. So the way this specific RNA prep is done is actually we would obviously have cDNA synthesis, right? We need to convert our RNA into cDNA, but then we would use that transposon that we use in Nextera library preps. We would leverage on transposon technology to actually create that library, so to fragment and tag our DNA, right? And this allows us to have that up to 384 unique dual indexes. So it allows us to have higher amount of data, so higher output of data out there, well, of samples that we are looking into. I will not go into too much details about how RNA prep with enrichment is working, but just to let you know that you could do exome enrichment if you want. Previously, we have TruSight RNA exome panel, and you will notice that it has many more steps than Illumina RNA prep with enrichment. And what Illumina has done is try to make the whole workflow 50% faster, and also to have 80% less hands times on, and also to have that single enrichment workflow, right? So to make it more accurate, and obviously this makes it more cost effective, right? When we're also talking about targeted approaches, I won't go into too much detail, but just to mention that we are having also amplicon-based approach, right? For those of you that are familiar with the AmpliSeq, and we have many different panels. Most of it will deal with some kind of a cancer research, but there are many different out there. But in these AmpliSeq panels, some of them are both DNA, so we will investigate DNA, but we will also investigate RNA, and we will look into specific transcripts, or we will look into specific fusions that we know are specific for that certain cancer type. So why is RNA sequencing so important in oncology and cancer research, and why we are talking about gene fusions when we're talking about this targeted approaches, either TruSight pan-cancer or TruSight RNA fusion or any of these amplicon panels, is because 80% of all human tumors have been shown to carry the translocation and gene fusions, right? And the majority of these fusions have a significant impact on tumorigenesis and have a strong association with morphological phenotype, right? So the menu of therapies will actually target this fusion. So when it comes to oncology, it's really important that we understand which fusions is a patient having, right? And this expression analysis across cancer samples can serve to identify driver genes and making them potential targets for that therapy, right? And that is really important. So where our RNA sequencing will help, obviously, not just to, we want to detect fusion and different alternative splice sites, but characterize that non-coding RNA and quantify allele specific expression, generate differential expression profiles, and identify drug response associations, right? And this will help us to discover, develop, and profile those pharmacodynamic biomarkers. The last application that I want to touch on, I'm not going to go too much into it because we will deal with it next week. But a really popular application that is popping out is single cell RNA sequencing, and here Illumina has partnered with Takara to make this possible. So we would employ a smarter ultra low kit from Takara, which can go which the input sample input concentration for the sample would be as low as 10 picograms of RNA. So we will use Takara smarter ultra low kit to actually create double-stranded cDNA, and then we will go into Nextera XT to create a library which then can be sequenced onto on Illumina platforms, right? And the reason why single cell is becoming such a hot topic is obviously we spoke about RNA being dynamic and being different all the time and being different between the cells. And obviously we need to understand which genes are on and off expressed, yes or no, and what level, right? And we need to understand the abundance, but also understand is there a different fusions happening in those cells. And just to finish it off, I just want to show you that in the past 2 days, previous week, and also today, we talked about many different applications, right? But it's all on one Illumina technology, right? It's utilizing those production scale systems, right? So regardless if we are talking about whole genome sequencing, right, either small microbes or human, ideally, or exome sequencing, or like we talked today about differential expression, fusion detection, single cell analysis, we have a single technology that allows us to do this multiple range of applications. And with that last slide, I would like to thank you for joining us today. If there is any questions, please let me know. And please do join us next week. Like Colette mentioned, we will go a little bit more into bioinformatics, and we will touch on specific pipelines that we can utilize to either do when it comes to DNA application or also RNA applications as well. Colette, are there any questions?
Thanks, Marija. I did see a question, and maybe just ask the team for the questions that came through.
Marija, there's a question for you. How important is the depth of RNA sequencing compared to biological or technical replicates for differential transcriptome studies in bacteria?
Okay. Yeah, so excellent questions. Those biological replicates are really, really important, right? And that is something that we need to have, right? Technical replicates we don't have to have because we are sequencing so much deeper. We can be confident that what we are getting is a true transcript, right? If you're sequencing very deep, but biological replicates we need to have, right? Because different samples might have a different levels of expression, might have, depending on what your biological question is, or what is the setup of your project, what is the treatment that you're giving to those specific samples, right? What are the conditions that you are exposing your samples with? Obviously, the expression will differ between those samples, and therefore it's really important that we have biological replicates. Are there any other ones?
She just also wanted to know whether we offer RNA sequencing services, which I was responding to, but you can also.
Yes, so separations, we don't offer any sequencing services. We help you with procuring your own NGS system, Illumina system, and providing you with the reagents to run these instruments, also library preps, but we are not service providers of sequencing as such. So we enable you to do NGS, but we don't sequence it.
Any other questions? You can unmute yourself if you would like to ask any questions. So if there's no more questions, ladies and gentlemen, I'd like to thank you for joining us today. As we said, next week, same place, same time, and we can take you through the data analysis with the Illumina data analysts per se. So you will also be in the hands of the experts on data analysis. Thanks so much for your time. See you next week.
Thank you. Bye.